हेलो स्टूडेंट्स एंड वेलकम। आज का हमारा जो टॉपिक है दैट इज इम्पोर्टेंस ऑफ सेल बायोलॉजी एंड इट्स एप्लीकेशन इन इंडस्ट्रीज. So first we'll complete with the topic that is importance of cell biology. Cell biology, it is the biologists who study about it, and in this field we completely study about that how which are the activities taking place inside a cell, like the metabolism like actually it is a combination of the catabolism and anabolism. Catabolism का मतलब है that is breakdown process, anabolism का मतलब है that is synthesis का process. जब भी carbohydrate जो है it is broken down into the glucose, so it is an example of catabolism. And the glucose, it is going to form the glycogen, so it is a kind of a synthesis process. So all these metabolic reactions which is taking place inside the cell is called metabolism. Even because when the cell divide, the growth takes place. Even if we talk about the stem cells are the cell which can give rise to any type of cell, that is called differentiation. Then inside a cell there is DNA which is going to act like a genetic material, and the information is going to get transferred from one generation to the another generation, that is because of heredity. And any kind of a sudden change inside the DNA which is present inside a cell, it leads to the evolution. So all these activities are going to be seen inside the cell, and we will study in the molecular level. So there are number of applications. There is a wide applications of cell biology, due to which it has given rise to number of biological branches. You can say these are the new hybrid biological sciences. So here one by one we are going to study about it. सबसे पहले आता है दैट इज साइटोटेक्सोनॉमी. साइटोटेक्सोनॉमी हम में क्या पढ़ते हैं, जो भी लिविंग ऑर्गेनिजम्स हैं दे आर गोइंग टू हैव सेल्स. So we compare the cells of the living organism. So we study about the chromosomes which are present inside a cell. जैसे कि कोई living organisms हैं like the potato, so the total number of chromosomes are 48. If we talk about mango, the total number of chromosomes are 40. The cat, the total number of chromosomes are 38. Then dog, the total number of chromosomes are 78. So here we are studying these organisms by observing the chromosome number in a cell, and we are going to compare them. So this is called as comparative study of chromosome. So whenever we know a definite number of chromosome in a plant or animal cell, that is in a species, we are able to distinguish between them. We are able to compare between them, and we get to know about the resemblance, कितने ज़्यादा close हैं वो, evolutionary कितने ज़्यादा close है वो, so we study about it. So that is why ये information को collect करके ही we are able to keep them in a taxonomy. So taxonomical position पे रखने के लिए it is essential that we should know the number of chromosomes in a particular organism, and that is how the taxonomic units it has been studied, and it comes into the cell biology. Next comes about the cytogenetics. In this we study about the genetic material inside a cell. Like the here we study the genetic material DNA. किसी भी organism में the genetic material RNA हो सकता है like and viruses, या तो फिर mostly it is the DNA. So here we study about the heredity, how the transmission of characters is going to take place from parents to the offspring. Then variation, there are variation seen from generation to generation because of the change in the DNA. Next, mutation, a sudden change. It may be because of environment, it may be because of the toxic material. So DNA में है जो है एक किसी भी तरीके का चेंज आता है, that is called as mutation. किसी भी तरीके का sudden change जो आता है, that is called as mutation. So mutation, it could be beneficial mutation, harmful mutation और neutral mutation. अगर beneficial mutation होता है किसी भी genetic material में, किसी भी cell में, तो it will lead to the evolution. अगर harmful mutation होता है then it will give rise to the death of the organism. और neutral mutation, it is not going to cause any kind of change. Next is phylogeny. Phylogeny का मतलब होता है we study about the evolutionary relationship between the organism. Then morphogenesis, here genetic material के ऊपर डिपेंड करता है कि किस तरीके की जीन्स प्रेजेंट है और कौन से जीन्स एक्सप्रेस हो रहे हैं, वैसे ही मोर्फोलॉजी जो है वो ऑर्गेनिज्म के अंदर देखने मिलेगी. एंड इवन अबाउट द इवोल्यूशन, सो दिस इज हाउ वी स्टडी अबाउट द साइटोजेनेटिक्स. एंड इवन जो वेस्टमैन ने जर्म प्लाज्म की थ्योरी दी थी, he explained about the transmission of heredity characters. So it's all because of the chromosomes which are present in the nucleus, even which we studied about the Mendel's law of inheritance, ये भी cytogenetics में आएगा. Even the concept of gene, अगर आपको जीन के बारे में पता है, what exactly the gene is, suppose this is the DNA, this is the DNA, so a segment of DNA is called a gene. सो जीन जो है हो सकता है वो एक्सप्रेस करें, हो सकता है वो एक्सप्रेस ना करें. सो जो भी जीन एक्सप्रेस करता है दे गिव राइज टू द आरएनए, एंड देन आरएनए दे गिव राइज टू द प्रोटीन्स, और प्रोटीन्स जो है वो आगे जाके एंजाइम्स में फॉर्म हो सकते हैं या हार्मोन का फॉर्मेशन करते हैं. मल्टीपल देर आर मल्टीपल मल्टीपल यूज ऑफ द प्रोटीन विच इज टेकिंग प्लेस. So this is how the gene express takes place. So here this is also studied in the genetics. And even if we talk about the how actually the synthesis of the heredity material takes place, जो DNA जो है वो replicate करता है, और इस तरीके से जो है रेप्लिकेशन द्वारा हेरिडिटी मटेरियल जो है सिंथेसाइज होता है. इवन द क्रैकिंग द जेनेटिक कोड, किस किस टाइप्स के जो है हाउ मेनी नंबर ऑफ जीन्स आर प्रेजेंट इन द डीएनए इज स्टडीड इज इन साइटोजेनेटिक्स. then the hybridization techniques, the superior qualities of crop varieties. So how the superior quality crop varieties it is produced, it's because of the recombinant DNA technology. So all these terms are going to be seen when we study about the cytogenetics. And also the genetic and phenotypic expression, genetic control of growth, then role of genes in organic evolution. Next is about the cell physiology. In this cell physiology, we actually study which are the activities which are taking place in the life. So, study of like activities like the nutrition, metabolism, excitability, growth, reproduction, cell division, and differentiation of the cell takes place. That is how all these activities are seen in a particular living organism life. So further, these cell physiological activities they are very much important. Why? Because these are going to conduct the physiological activities at a cellular level. So इसी तरीके से हमें पता चल जाता है कि कौन सी फिजियोलॉजिकल एक्टिविटीज जो है नॉर्मल है किसी लिविंग ऑर्गेनिज्म में, या कौन सी फिजियोलॉजिकल एक्टिविटी अबनॉर्मल है किसी भी लिविंग ऑर्गेनिज्म में. सो बाय ऑब्जर्विंग दीज लाइफ एक्टिविटीज वी आर एबल टू डिस्टिंग्विश बिटवीन द नॉर्मल एक्टिविटी एंड abnormal activity. Next is about the cytochemistry. And this is also a branch of cell biology where we study about the chemicals which are present inside the cell, like the carbohydrates, lipids, proteins, nucleic acid, organic and inorganic chemical compounds. They all are present inside a cell. So this is what is called as biochemistry. So जो भी researchers हैं इन्हीं biomolecules को जो है आसानी से isolate कर सकते हैं, उनको analyze कर सकते हैं, और कौन कौन से मॉलिक्यूल्स किस किस ऑर्गेनल्स में प्रेजेंट है. ऑर्गेनल्स लाइक द राइबोसोम्स, माइटोकॉन्ड्रिया, एंडोप्लाज्मिक रेटिकुलम, ऑल दीज आर ऑर्गेनल्स. सो कौन कौन से केमिकल कंपाउंड्स जो हैं ये ऑर्गेनल्स के अंदर प्रेजेंट है, और किस तरीके का फंक्शंस करते हैं सारे ऑर्गेनल्स इन केमिकल कंपाउंड के हेल्प के द्वारा. सो वी ऑल स्टडी इन द साइटोकेमिस्ट्री. Next is the ultrastructure and molecular biology. It deals with the ultrastructure of cell organelles. जो organelles हैं, cell organelles like the endoplasmic reticulum है, lysosome है, mitochondria है, how exactly the structure look like the of organelles inside a cell, how the cell membrane do look like, जो plasma membrane है वो किस तरीके से दिखाई देती है, उसका structure कैसा है, उसका composition कैसा है, हम ये सब स्टडी कर सकते हैं इन द मॉलिक्यूलर बायोलॉजी बाय यूजिंग द इलेक्ट्रॉन माइक्रोस्कोप. Electron microscope it helps to study all the organelles which are present inside a cell. So बहुत ही important ब्रांच है सेल बायोलॉजी की, जिसकी वजह से वी आर एबल टू स्टडी इन डिटेल अबाउट द सेल बायोलॉजी. सो वी स्टडी अबाउट द बायोकेमिस्ट्री, फिजियोकेमिस्ट्री, मैक्रोमोलिकुलर एंड कोलॉइडल केमिस्ट्री. इट्स ऑल बिकॉज ऑफ द इलेक्ट्रॉन माइक्रोस्कोप. Then here this knowledge of ultrastructure of the cell is of fundamental importance because practically all the function and physico-chemical transformation takes place with the molecular architect of the cell at the molecular level. सो आज हम बात करें अबाउट द रीसेंट डिस्कवरीज, जैसे कि डिस्कवरी ऑफ द मॉडल ऑफ डीएनए. सो हियर इट इज also the kind of information which is helping us to know about the biomolecules which are present in the inside a cell. जो डीएनए है वो किस तरीके से दिखता है, किस तरीके से काम करता है मतलब डीएनए जो है, इट इज प्रेजेंट अ नंबर ऑफ जीन्स. जीन्स जो है वो एक्सप्रेस होंगे तभी आरएनए बनेगा, और वो here further RNA, the flow of information from RNA to protein is called as transcription. So ये जो processes हैं, transcription है, even the translation and flow of information from DNA to RNA. So all these processes are being studied in the molecular biology. So here the model of DNA, how the DNA look like, then how the protein formation is taking place, the genetic code, all these are the concept which we studied in the modern biology, that is in the molecular biology. Next is cytopathology. In this cytopathology, it is actually a branch of pathology where we study about the how we are able to diagnose, how we are able to identify the disease at a cellular level. So here also there is a use of the molecular biology. So here to the pathological science which help us to understand various human diseases at the molecular level. So most of the diseases जो होते हैं, they are seen, it is because of the disorder seen in the genetic code, that is in the DNA. सो अगर कोई जीन है, इफ इट इज इन डिसऑर्डर्ड कंडीशन, नॉन फंक्शनल कंडीशन, तो वो आरएनए नहीं बना पाएगा, आरएनए बनेगा तो प्रोटीन नहीं बनेगा, प्रोटीन नहीं बनेगा तो एंजाइम्स का फॉर्मेशन भी नहीं होगा. सो इट इज गोइंग टू हैम्पर द मेटाबॉलिक एक्टिविटीज इनसाइड द सेल. सो इट विल लीड टू नंबर ऑफ डिसऑर्डर्ड इनसाइड द सेल. So there are number of toxic chemicals, there are number of viruses which are going to create abnormal activity inside a cell, inside a tissue. Like toxic chemical, they can give rise to the cancer condition, and viruses, they also affect the cells and the tissue. So we are able to show a close relationship between the cytology and pathology on hand in hand. And medicine, it is one of the another hand which in turn useful to understand the various diseases of animals. Next is cytoecology. ऐसा कहा जाता है कि साइंस दिट्स साइटो इकोलॉजी, इट इज अ साइंस इन विच वन स्टडीड अबाउट द इफेक्ट ऑफ इकोलॉजिकल चेंजेस ऑन द क्रोमोसोम नंबर. ऐसा कहा जाता है कि इवन एनवायरमेंट कैन ऑल्सो प्ले अ रोल इन चेंजिंग द नंबर ऑफ क्रोमोसोम. सो द साइटोलॉजिकल स्टडी, we study of plants, we study of animals, and here it is revealed that ecological habitat and geographical distribution, they also affect the correlate with the number of chromosome. Next is cytology. Cytology, it is a branch of biology that studies the structure कैसा है किसी भी सेल का, प्लांट का या एनिमल सेल का, कैसा फंक्शन करता है कोई भी सेल, प्लांट या एनिमल सेल. सो जैसे कि एक एग्जांपल लेते हैं, सपोज दिस इज अ ह्यूमन ब्लड सेल. सो देयर आर नंबर ऑफ टाइप्स ऑफ ह्यूमन ब्लड सेल, erythrocytes, RBCs, then platelets, then WBCs. They are having some different structures, they are going to have some different functions. So this study is going to come into the example of cytology. Next is Cyto Evolution. There are some energies which produces some radiations, ionizing radiations like X-rays, gamma rays, and even if I talk about cobalt 60, they are very much harmful. They can cause a change in the number of chromosome. They can cause a change in the structure of the chromosome. If there is a change in the number of chromosome, then there is going to be seen a change in the morphology also. मोर्फोलॉजी भी जो है वो क्या हो जाएगी, किसी भी ऑर्गेनिज्म की अल्टर हो जाएगी, चेंज हो जाएगी. इवन इट कॉजेज अल्टरेशन, चेंज इन द फिजियोलॉजी ऑफ ऑर्गेनिज्म ऑल्सो. सो इट कैन लीड्स टू द म्यूटेशन and will lead to the evolution. Next is cell culture. Cell culture जो है, here first of all we study the cell in the lab, that is why it is called in vitro. So actually we study the cell biology, we understand the basic concept of the cell biology, like how the DNA duplicates, that is DNA replication, how the flow of information is going to take place from DNA to RNA, that is called transcription. Then how from the RNA protein is going to synthesize, that is called translation. So how the protein it is formed, Then energy metabolism, how the ATP, adenosine triphosphate, it is going to release the energy. Then receptor studies, हर cell के पास जो है there are some receptors present. So if I here talk about a cell, on the cell surface there are some receptors present, due to which these receptors, they are going to they are going to receive some signals, they are going to receive some hormones, and that is how the receptors, they help to receive the signal, and accordingly the cell is going to act. So there are receptors study also done. कुछ receptors होते हैं जो प्लाज्मा मेम्ब्रेन के ऊपर होते हैं सेल के, कुछ receptors होते हैं जो न्यूक्लियस के अंदर होते हैं सेल के. So this is how the receptors are present on the cell. Cell analysis, then cell division, there are various types of cell division like mitosis, meiosis, then gene expression. अगर flow of information हो रहा है from gene to you can say from DNA to RNA, RNA to protein. So this is called as gene expression. Then signal transduction, how the cell is going to communicate with another cell, that is called signal transduction. Apoptosis means the death of the cell. Effect of radiation, there are effects of radiation seen in the cell. Then population kinetics, then cell cell and cell matrix interaction, किस तरीके से एक सेल दूसरे सेल से कम्युनिकेट करेगा, या कोई भी सेल है मैट्रिक्स के साथ किस तरीके का इंट्रैक्शन है. सो मैट्रिक्स मतलब सेल के आसपास जो भी प्लाज्म है, दैट इज कॉल्ड मैट्रिक्स. Then even in the cell culture we study about the understanding about the disease processes, especially like the malignancy, that is a cancerous condition. Isolation of viruses, how the virus can be isolated from the tissue, from the cell, how the vaccine it is produced. Then production of biological important molecules. Even there is also a process that is cell fusion technique is also it is understood, it has been understand. So we study about the somatic cell genetics. Even we also study about the gene mapping. Gene mapping का मतलब है here suppose this is a chromosome, suppose this is a chromosome, so there are some genes present. So कौन से genes कौन से chromosome में present है, किस location में present है, chromosome के P arm में present है, chromosome के Q arm में present है, किस location में present है. और अगर genes हैं तो एक दूसरे की जीन बीच में कितना distance present है, जितने भी genes है उनके बीच में distance कितना present है, that is called as gene mapping, which are present on the chromosome. And also development of monoclonal antibodies which work in the immune system. Even we study about the gene transfer studies, there are transfer of genes from one organism to another organisms, that is here we have already studied in the recombinant DNA technology. So that the, if we take an example of gene transfer studies, if we have studied अपने बैक्टीरिया का नाम सुना होगा, that is called as BT. बीटी का मतलब है दैट इज बैसिलस तुरंजिनेसिस. बैसिलस तुरंजिनेसिस का जो है ये एक बैक्टीरिया है, इट इज अ बैक्टीरिया. इस बैक्टीरिया से जो है क्राई जीन को जो है रिमूव किया गया था, देयर वाज अ क्राई जीन व्हिच वाज रिमूव. ये जो क्राई जीन है इट हैज बीन नाउ इंट्रोड्यूस इनटू द प्लांट्स, इट हैज बीन इंट्रोड्यूस इनटू द plants like the cotton plants, like the cotton plants, that is why we also call these bt cotton plants. So whenever any kind of worm, suppose any kind of ball worm, when it eats these kinds of plant, here the cry gene, here the cry gene, here it get entered inside these organisms, and now in these organisms the protein it is produced, and that protein it is a toxic protein. This is a toxic protein which is going to get produced, and here these toxic protein in these ball worm can cause death of these worms, because these toxic proteins, they cause pores in the gut of these insects, in the gut of these ball worms. So that is how the plant it becomes the pest resistant. So that is why this is a beneficial use of the gene transfer studies. Even we studied about the stem cells culture, how any kind of non-functional cells, they can be replaced by the functional cells. So here this is how we studied about the stem cells, and here these are the applications of the cell biology. Cell biology, it has also been used in industries. Like we study about the cell, it is a structural, fundamental, and functional unit of living organism. जैसे atom होता है, it is a fundamental unit in chemical structure. So this it can be considered as an organism in itself. Cell जो है वो खुद में ही एक organism है जिसके अंदर बहुत सारे elements present हैं, or it is very much important, it is a significant which is present inside the organism. So in our advanced knowledge of the cell, it has produced a fundamental change in the interpretation of the cellular structure. Just because of the depth study of the cell biology, we are here, we have a kind of a good information, we are having a good interpretation related with the cellular structure. So हम सेल को जो है बहुत ही अच्छी तरीके से पढ़ते हैं, उनका अल्ट्रा स्ट्रक्चर क्या है, कौन से large repetitive units हैं. It is being studied with the help of the microscope techniques. Even if we talk about the electron microscope, so it has contributed a great information in the cytology. So here the cell, it has been studied from various aspects like morphology, biochemistry, physiology, genetics, development, and molecular structures, which help us to understand all the activities which are taking place inside the cell, like the metabolism, growth, differentiation, heredity, and evolution. सेलुलर लेवल पे भी हम इनको पढ़ते हैं और मॉलिक्यूलर मेटाबॉलिक लेवल पे भी हम इनको पढ़ते हैं. So this is how our lecture ends, and thank you very much.